So today we're going to talk about the structure of nucleic acids, specifically the structure of DNA, in a little bit greater detail. So as you probably know, nucleic acids are polymers. They're made of these repeating elements, and the monomers, those repeating elements in nucleic acids, are nucleotides. And there's a picture down here of one nucleotide, and you can see it has kind of three different components: this piece, this one, and this one over here. Each nucleotide consists of a pentose sugar. So pentose just means it makes this five-sided ring. In DNA, this sugar is called deoxyribose. In RNA, it's a different sugar called ribose, but here it's deoxyribose. One important thing to note, as you may remember, sugars or carbohydrates are mainly a ring of carbon molecules. Each corner of this represents a carbon. And in these rings, the carbons are numbered. And so remember, every place that there is not a molecule shown here, there's a carbon. Or sorry, not an atom shown. There's a carbon. There are also lots of hydrogens that are not visible in here. Everywhere, for example, this carbon has 2 bonds shown here and here. Carbon needs to form 4 bonds, so that means there would also be 2 hydrogens attached here. They are not drawn on here because that would just make this drawing much too difficult to look at. But the important thing right now is to notice that the carbons in this sugar are numbered. And the way you read these numbers is you read that little asterisk up there as a prime, as the word prime. So we say this is the 1' carbon, 2', 3', 4', 5'. And that's going to be important when we start looking at how DNA functions, because you can see the other pieces of the molecule that are attached are different. The phosphate group is attached to the 5' carbon, and there's this hydroxyl group that's attached to the 3' carbon. And this is important because this hydroxyl group off the 3' carbon is where this nucleotide is able to bond to the next, which we'll talk about in the next slide. So you do need to know which way these are numbered. I kind of remember that the 5' is the one that hangs off by itself. There is also a phosphate group, like I said, over here, hanging off that 5' carbon. And phosphate groups, remember, are negatively charged. That's an important thing to understand because there's so many phosphates as part of a DNA molecule that overall DNA has a negative charge, mainly because of all these phosphate groups. There is also a nitrogenous base, which is those C's, G's, A's, or T's. If we're talking about RNA, it could also be the base uracil, the U. The bases are divided into 2 different groups. There are bases called pyrimidines. This is an example of a pyrimidine. The way that you can recognize them is that a pyrimidine just has a 1-ring structure, as opposed to the other group, the purines, which have a double ring structure. So let me put that up here. So the purines are guanine and adenine. The pyrimidines are cytosine, thymine, and uracil. And here's an example that might help you remember which is which. We talked about this before. This at the top is a molecule of ATP, adenosine triphosphate. But remember, we talked about the fact that this is actually a modified DNA nucleotide. And so this right here, because it's adenosine triphosphate, this is the base A. And you can see it has this 2-ring structure that's indicative of a purine. Purines have that double ring. Pyrimidines just have the single ring. As we go through and talk about this today, you may want to grab the worksheet from yesterday, the chemical structure of DNA, and add some notes and make sure that you appropriately labeled all the different parts of your molecule as we go through here, because I am going to ask you to go back and finish up that sheet if you haven't already. So you can use today's lecture to help you complete that. So these nucleotides made up of a sugar, phosphate, and base link together into long chains. They're the monomers that build this larger polymer. And so in this picture here, you can see, as with many polymers, these are joined together through dehydration reactions. And in this case, the type of bond formed through dehydration reactions is called a phosphodiester bond. And on the chemical structure of DNA worksheet, I told you that we would label this one together. An ester bond is a chemical term that refers to this type of bond right here. That forms between this carbon and oxygen. And so you can see as these nucleotides link together, we actually get two of those. That's the diester with a phosphate in the middle. So a phosphodiester bond is actually this entire section. It's that bond between the carbon and oxygen, the entire phosphate, and that other bond between the carbon and oxygen. That whole thing in the middle there forms that phosphodiester bond that links these nucleotides together. So you can see the sugar is up here and the phosphate's here. The base in this diagram is just summarized with the letter. So there's your cytosine, guanine. That's what's kind of pointing towards the middle of that DNA ladder. So as these join together, that's what creates that sugar phosphate backbone along the sides of the molecule. So here we have the sugar, that's that 5-carbon sugar deoxyribose. Here's your phosphate group. Here's the next sugar. And that would then link to the next phosphate group down here. So you get that backbone all along the side with the bases kind of hanging out here in the middle. One important thing to note is because of the structure of those nucleotides, this chain that's formed along the side has directionality. The two ends of it are different. At the top end in this picture, we have the 5' carbon that would be attached to the next phosphate group up here. But then you can kind of follow this chain, then the 3' carbon links through that phosphodiester linkage to the next 5', which goes down and links. So this right here is the 3' end of our chain, because the 3' carbon is the one where the next bond can form. So we call this the 3' end of the chain, and this one is the 5' end. An entire molecule of DNA, of course, is formed out of two of these polynucleotide chains. Polynucleotide just means many nucleotides. So on the previous slide, we were looking at that phosphate, sugar, phosphate, sugar. There's that backbone through there. And you have the same thing over here. We have sugar, phosphate, sugar, phosphate. So we have that sugar phosphate backbone on both sides with the bases hanging out toward the middle. The two polynucleotides link together because of hydrogen bonds that form between the bases. And you can see they follow the base pairing rules, A with T, C with G. You can also see that a purine and a pyrimidine always bond together. There's always a 2-ring and a single ring that keeps the distance equivalent all the way down that chain. The A and T can form 2 hydrogen bonds, and the C and G can form 3 hydrogen bonds because of the chemical makeup of those bases. You'll also notice because of how those bases are able to form those hydrogen bonds and the way that they need to be oriented to do that, these strands are what is known as anti-parallel. And I'll uncover the rest of that so that you can really see this. Again, if we think about the directionality of these strands, this is our 3' end of this strand, and this is the 5' end. But the opposite strand goes the other way. Here's our 5' end, and here's our 3' end. And you'll notice the arrows go in the direction, they go towards the 3', and that's important. That will be important as we start talking about how we replicate and build DNA, because the enzymes that build DNA are only able to add on to the 3' end of a strand, because they can only form a bond using this hydroxyl group. So we will talk about that a lot more as we get into DNA replication, but that's why the arrows are pointed towards 3'. That's the direction this strand could grow, and this strand could grow in this direction. And then, of course, these strands twist up together to form that double helix shape that we are so familiar with. So there are a couple of tasks that I would like you to do today to kind of finish up this topic. First of all, if you have not already, please make sure you finish up that chemical structure of DNA sheet. All of the labeling and coloring should be done. And there are a few questions at the bottom. Make sure you answer those and keep that sheet with your notes. That's kind of part of your notes on this topic. Then there is also a modeling activity. We are going to next be talking about the process of DNA replication and how DNA copies itself, which is very closely related to its structure. That's part of the reason that the structure of this molecule is so important. Today, you're just kind of doing some introduction activities to review a little bit of what you already learned about DNA replication and start familiarizing yourself with some of the enzymes involved. That's a lot of the new pieces that we're adding are a bunch of different enzymes and their jobs. So for today, you need to go in. There are some instructions on the post in the homework section. You need to complete the first 3 slides on that activity today. We'll be finishing the rest up next Monday after we finish the lecture on DNA replication. One other note that I just wanted to point out to you, there will be an assessment tomorrow, so watch for that post. Tomorrow's your last school day of the week. Tomorrow's assessment will mainly be on cellular respiration and photosynthesis. There will not be questions about DNA yet. This will be on the assessment that you take next week after we've gotten a little further into this. So make sure you study respiration and photosynthesis and contact me if you have any questions. Have a great rest of your day.